Good evening everyone, so welcome to my YouTube channel. So here today we are going to be looking at our part 2 of the cell membrane. So some parts of the cell membrane have got ligand-gated ions. So now what are these ligand-gated ions? So a ligand is simply a chemical substance that is sent and binds to a receptor. So the receptor might be made of glycoproteins or glycolipids. So when the ligand binds to a receptor, it may cause the opening of the protein channel or the closing. So now when we look at our ligand-gated channel, so this is how the mechanism is. So this is one of the examples of what we are going to call facilitated diffusion. So let's imagine our red dot is our ligand. Remember, a ligand is simply a chemical substance that binds to a receptor. So it's a fancy way of just saying a chemical substance that binds to a receptor. Then it causes the change or the closing, the change of the protein channel by either opening it. So when it opens it, it allows certain substances to pass through. So this is the way it is. So this red dot is the ligand. It goes like that. Then it goes to our receptor. Once at the receptor, it will cause, so as you can see, our protein channel here is closed. So when the messenger, which is our ligand, comes there, it communicates with the cell, thereby allowing the cell to be able to open up. When it opens up, it allows specific substances to pass through. The examples of where ligands are very, very much applicable is when we look at our neural transmission. So when you look at neurons, we have from one neuron to the other. How come that the impulses are able to pass through and it's on the impulses? This is where ligand-gated channels are important. The ligand will be able to bind to the receptor like that, thereby causing the protein channel to be able to open up to allow the electrical impulses to pass. So the example of ligands that we might have there are the neurotransmitters. Neurotransmitters, we might have acetylcholine. So this is an example of a ligand that is associated with the transmission of impulses, or dopamine. So this is an example. So now ligand-gated channels are very, very important. So our ligand-gated channels might have different shapes. Some ligands may be triangular, so they will be able to stick to the surface of the receptor like that. So this is our receptor, they stick like that. So as you can see, ions, when the ligands have not more like attached themselves here, it will not be able to open up, therefore the ions won't be able to enter. But as the ligand attached there, the ions are able to enter like that. So that is the importance of the ligand-gated channel. As we can see, we have a ligand. If it is not attached, ions might more like move away from the cell. They might go like that, then bounce from the cell. So this is when the ligand binds substances, the channel will open and the substances will be able to enter. So the substances that enter are very, very specific. So this is our neurotransmitter. We have the ligand-gated channel there. It is open because the ligands are attached at that point. So to simplify things, this is the notes that I've created for you. So a ligand-gated channels are membrane proteins. So they are membrane proteins, which are ion channels that open and close in response to the binding of specific molecules. The specific molecules are the chemical substances which we were calling ligands. So when open, they allow specific, it's not just anything, specific ions to move across the membrane down their concentration gradient. So it is down their concentration gradient. Then it is also an example of facilitated diffusion, meaning to say for the ions to pass, they need some sort of a channel, which we are going to call our protein channel. So that is how the ligand-gated channel operates. Thank you very much for watching. See you in the next tutorial.